we're trying to understand the genetics of a cancer, what we normally do is to take a cancer and sequence that and look for the genetic changes in that cancer. What we've done differently in this study is to actually look at the mutations that are present in normal tissue to see the very first seeds of cancer being sown.
We think that cancer starts from a single cell that picks up a cancer-associated alteration in its DNA. And then that cell can divide and create a clump of cells which carry that same altered DNA. And if that clump of cells persists in the tissue for long enough, it can pick up more and more mutations. And so eventually it picks up enough mutations to cause a cancer.
What we did in this study was to take skin from 4 normal individuals, aged between 50 and 70. And from those 4 individuals, we took several hundred very small biopsies of the skin, no more than the size of the head of a nail. And we sequenced those across the genes that are known to be involved in skin cancer. What we found was that there are a huge number of genetic changes in normal skin cells. So we can see hundreds of these per square centimetre of normal skin. It looks like these mutations are accumulating at an average rate of close to 1 mutation per cell per day. What is perhaps even more surprising than the sheer burden of mutations was that in amongst them were all of the mutations that we might expect to see in skin cancers.
Sunshine changes DNA in a particular way. It's as if it signs the DNA with a tag. So we know when it's sunshine that's caused a change in DNA. And what was striking in this study is that the changes that we saw in the DNA were almost all due to sunshine. Some of those changes were in genes that show up in cancers, but the chances of that cell turning into a cancer are extremely, extremely low. All mammals that live outside, that would be most of them, have evolved over millions of years to deal with the fact that sunshine changes DNA in the skin. And what we're very keen to understand is the defences that we have that stop all of these cells carrying potentially cancer-associated mutations developing into cancers.
What we see in these normal tissues is that the clusters of cells are beginning to expand. They've expanded. Maybe there are twice as many cells from a clone of cells that carries a cancer-associated mutation compared to cells that don't. What that's telling us is that they're taking the first steps on this journey towards the out-of-control proliferation that you see in cancer, but something's constraining them. And potentially this approach gives us a route into understanding what those mechanisms might be.
So what does this study mean for most of us? Well, what we've seen is that in normal skin, we've got large clumps of cells with genes that can show up in cancer. Where have those genes come from? They've come from sun exposure, which we might have had years before. So avoiding excessive sun exposure when you're young is a really good thing to do. Now, some older people will think this doesn't apply to them, but actually all of the patients that we got skin from for this study were in middle age. And once you've got middle-aged skin with these areas, these clumps of cells carrying the changes in DNA, it's really good to avoid adding extra DNA changes to those because it's the further changes that can result in cancer.
So what I'm really excited about in this research is that we can now scale this up across all sorts of different questions with different organs, different people, different ages, different exposures, and this will give us a much deeper understanding of how cancer begins.